All right, everybody. We're back about to do part two to this basic chemistry. Last time we talked about atoms, molecules, compounds. We talked about ions, talked about ionic bonds versus covalent bonds. We talked about acids, bases, and pH. We just really basic, man. Just keeping it really basic because what we want to do is we want to take that material and then build off of it for the rest of the semester. What I want to do now is I want to kind of tackle with part two and talk about organic molecules. Talk about some organic compounds. Specifically, I want to talk about carbohydrates, lipids, proteins, and nucleic acids, and really just get into that and relate it to the human body, talk about some basics with that. And then as we go on through the next two semesters, really build on that and get a little more complex. So without further ado, let me just go ahead and skip to the screen share mode and go ahead and jump into this. And I'm gonna go ahead and pull up the notes here now. So let's talk about this. Biological macromolecules, general characteristics about the four groups, four classes of organic molecules. The difference, one of the major differences between an organic molecule and an inorganic molecule is that an organic molecule contains carbon. All right. When I was in school, they taught us that organic molecules contain carbon and hydrogen, but that an inorganic molecule cannot contain both. Um, I don't see or hear that taught anymore. Typically, what they tell people is that an organic molecule is one that has carbon in it, just like you see fatty acids having a carbon chain backbone to it, um, whereas an inorganic molecule does not contain carbon in its makeup. So, what we're going to talk about today, we're going to focus the next 15-20 minutes all on organic molecules. And so we're going to start off with lipids. Typically, when I talk about this, I start with carbohydrates, but we're going to take a twist. Hey, let's do something new. Let's go with lipids. So lipids, are very interesting substances. They are water insoluble, which means that if you stick fat in water, it's not going to mix. If you've ever fried something in a frying pan and it leaves some oil in there, like let's say that you fried something in some butter, or you put a little type of fat in the pan and you fried that food in it. When you stick that pan in the sink with some water, the fat will float on top of the water. It does not mix together. Water and fat don't mix. Okay, so that means that when it doesn't mix together, it's insoluble. If it does mix together, then it's soluble. And generally, we see that lipids can function as stored nutrients, cellular membrane components, and hormones. And this is just a quick and easy way to say, hey, this is what these things are called when they are they're carrying out these functions. So let's talk about the first one, which is a triglyceride. What I'm doing is I'm matching the class with the function. Stored nutrients are usually stored as triglycerides. Cellular membrane components are usually referred to as phospholipids, and hormones are steroids and eicosanoids. So let's go ahead and jump into this really quick with the triglycerides. Uh, excuse me. That was weird. I had a hiccup and a burp at the same time. That's scary. If you notice the little corner over there, everybody can tell that that's a cartoon. However, the average person these days has no clue what in the world that cartoon is. I go old school with cartoons. That is a clip from a Transformers episode. Not the Transformers that these kids are watching now. This is nothing compared to the old school stuff back in the day. And those Transformers right now, they are stealing energy from the Earth and creating what are known as energon cubes. And energon cubes back in Transformers day were units of energy that could be stored and then pulled out and utilized by any transformer at any given time. Triglycerides are very similar. Triglycerides are units of stored fat, stored lipids that your body can pull out when necessary to actually use. It's used as long-term storage, and the good news is it's really good in energy. As a matter of fact, 1 unit of triglyceride, 1 molecule of triglyceride is worth 2 molecules of sugar. So as far as the amount of energy that you get out of a triglyceride is absolutely wonderful. There's just one problem with it. It's used for long-term energy, and your body likes to store it, and it actually uses energy to have to break this thing down and pull it out and use it. It's kind of like using your body using a triglyceride versus using sugar molecules is the same as finding a pack of batteries still in their package versus a couple of batteries sitting on a table. Now, of course, that pack of batteries has 4 AA batteries in it. They're fresh, they're new. No one's opened that package. Those 2 AA batteries sitting on the table, you know that those 2 batteries are new. They're old, and so they don't have much juice left. But if you're looking for something to pop into your remote control, more than likely you're going to pick up those 2 batteries off the table, because opening up that pack is going to require you to get a knife or scissors or something sharp. You're going to have to use your teeth. You're going to bite into the pack. You're going to cut your mouth. You're going to be mad. You're going to throw it across the room. You're going to try to tear that thing open. And it's going to require work and energy to get into there to get those 4 brand spanking new batteries, while just going over and getting those 2 old batteries to use. And that's the difference that your body sees in using a triglyceride versus a sugar molecule. So your body says, hey, I'll use the sugar molecule and I'll just store the triglyceride. And where you store the triglyceride depends on 2 things. It depends on it depends on 3 things. It depends on your lifestyle, it depends on your genetics, and it depends on your gender. Because your lifestyle will have a lot to do with where you store those triglycerides. The more active you are, the more of a metabolic rate your body will have, and the more of those triglycerides your body can get used to burning. Your genetics will play a large part in that. If everybody in your family is plus size, then there's a strong possibility that genetically you might be a plus size, a full-figured woman, or full-figured man. I come from a grove of trees. Like, everybody in my family are tall and thin. It has nothing to do with us eating right. I can assure you that's not the case. It's purely genetics. Everybody's tall and thin. It could have easily been the other way around. My genetics could have had very large individuals in my family, and so we could have been really tall, really football player looking guys, but we're not. And the other thing is gender. Women put triglycerides, they store triglycerides in different areas of their body that are totally different from where men store their triglycerides. Women, their triglycerides can build their body to make like to start shaping like a Coca-Cola bottle, and we're all happy for that. For the fellas, we start storing triglycerides, and one of the first places it goes is to our love handles, and it goes into the front of our stomachs. So you'll see men oftentimes battle this as they approach later on in later years. They start packing on weight, and they're still muscular, but the weight seems to want to all go to the belly. So this is a gender issue. So these triglycerides, when they are packing into our body tissues, we refer to these tissues as adipose tissue. Adipose tissue is formed from adipocytes, which are actual cells, and the cells actually store the triglycerides in their inside of themselves. They actually store the triglycerides in their cytoplasm, which is why when you look at adipocytes, they have this yellowish tint to them. It's because they're storing the fat literally in the cytoplasm itself. Now, don't knock triglycerides when it's stored as adipose tissue, because it can be used as structural support, cushioning, and insulation. I mean, adipose tissue. Did you know that you have adipose tissue behind your eyeballs? You have adipose tissue behind your eyeballs so that it's a cushion, because when you ride a roller coaster, or when you're riding you ride a mountain bike, or you you do swimming in some rough waters in the ocean, or you take off and you land in these commercial airliners, there's a lot of stress that goes on your eyes. And the adipose tissue behind your eyeballs helps to cushion your eyeballs to reduce damage. Um, of course, it can work as structural support. You have adipose tissue that is wrapped around certain other soft organs that act as structural support in your abdominal cavity, and it acts as insulation, which ain't half bad because it keeps you warm. Us skinny guys, man, we miss that adipose tissue, and we can be kind of cold at times. So I remember my first time I ever went to Chicago, man, those winds coming off those Great Lakes, they cut right through you like a hot knife through butter. So when we build triglycerides, it's known as lipogenesis or lipogenesis. When we break down triglycerides, it's lipolysis. So lysis means to break, genesis means to create. Here's a basic image of a triglyceride, tri meaning three, glyceride referring to the glycerol backbone. You can see over here this purple box, this purple rectangle. It shows the glycerol backbone. That is a separate molecule. And you can see the three fatty acids that are attached to the glycerol backbone. The glycerol backbone is essentially a molecule used to help carry the three fatty acids around to wherever they're going to actually go. And so what your body does is when it goes through lipogenesis, you actually take the glycerol and you attach the fatty acids to it, and then you create that triglyceride. But if you do lipolysis, then you do a process called hydrolysis. And in hydrolysis, what occurs there is you can see the process right there. You notice the hydroxyl groups that are here. There's a hydroxyl group. There's a hydroxyl group. So in dehydration, what you're doing is you're taking the water out. So you're snatching a hydrogen. You're snatching this OH group, and that causes this this bond to form with this double bond on the oxygen and the carbon linking to the oxygen here. But if we put the water back and through hydrolysis, we add the water, and so a hydrogen from the water attaches here. The OH from the water attaches there, and then we finally have the fatty acids separated from the glycerol backbone. And now we can use the fatty acids, which are great for energy. We can actually use the fatty acids. And a lot of times what we'll do is we'll take those fatty acids, these three chains, we can take those fatty acids and we can stick them in a mitochondrion inside of a cell, and we can burn those fatty acids as energy. And we get some really good energy out of that. Get a lot of ATP for the cell to use so they can run. Another form of lipids is a phospholipid. You'll hear later on when we get to the cell chapter, we'll talk about the fact that the cell membrane has a phospholipid bilayer, which in other words, this is a phospholipid bilayer, this image right here. And you'll oftentimes hear them talk about, that a phospholipid has one end of the glycerol. Same thing like a triglyceride, it had a glycerol backbone. It has one end of the glycerol backbone with a polar phosphate group with another organic group, and it talks about the fact that the glycerol, the phosphate, and organic group is known as the hydrophilic head. The fatty acid group is nonpolar, known as the hydrophobic end. Here's the hydrophilic end, these round circles, and then these are the hydrophobic ends, which are here. Now you got to remember that a phospholipid is a fat. It's a lipid. So when you're hydrophobic, you're not friends with water. All right. Because of that, that means it makes it difficult for water-soluble substances to pass through this phospholipid bilayer. So, a water-soluble substance like a protein will not pass through this phospholipid bilayer unless you have a special carrier mechanism that's going to bring that protein in or let that protein out. So if it's something that's lipid soluble, something that's some type of fat, it can pass through this wall because this wall is made out of fat. It's a double layer of fat, and fat mixed together very easily. So anything that's fat based, fat soluble, lipid soluble, it can pass through the phospholipid bilayer, go in and out of the cell as it pleases. But if it's water soluble, it's not going to pass through this wall so easily. And that's good because, you know, the cytoplasm, the cytosol inside the cell where everything's floating around is, you know, made up of water itself. So, you know, you don't want the water inside the cell to just spill out. And the cell is probably surrounded by an extracellular fluid or interstitial fluid, which is surrounding all those cells, and you don't want the water that's surrounding the cell to go into the cell without your permission. So this phospholipid bilayer is perfect. It keeps the water in the cell, keeps water out of the cell, and you know, it separates everything from the inside and the outside. It keeps the cell alive and safe. Another group of lipids happens to be steroids or chemical messengers. Steroids, you could go, man, you could talk about these things all day long, and I'm not. We're going to wait until we get further along to even delve into steroids. But basically, you know, a steroid, you've heard of them like testosterone and estrogen or steroids. They are specific types of lipids who they're not like your branched friends. You know, when we looked at a triglyceride, notice that there's a glycerol backbone, and then there's this line here and a line there and a line there. Very basic. And then we looked at a phospholipid bilayer. There's lines here and a little connector here. Lines there and a connector here. Everything's pretty straight. You get to like a steroid, no. Steroids have a ring structure to them. If you for the ladies out there who are using birth control, next time you go to pick up your birth control, ask for the instruction pamphlet and ask them if at the pharmacy, if because they can give you, sometimes they'll give you the stuff for free. Ask the pharmacy if they have the actual nomenclature for what steroid is in your birth control. And they may be able to ask them if they have the actual structure. Ask them if they have the actual chemical structure there. And you'll notice that if it's a steroid, it's made up of rings. It's a really big molecule drawn out on a piece of paper, and most of it is made up of little rings, five carbon rings, and little 6 carbon rings in there. There's all these little rings, and they're all connected to one another. And the cool thing about steroids is that because they are a type of lipid, as a hormone, when they go into the cell, if you look at this picture down here, as that steroid hormone goes into the cell, notice that there's no door here, there's no secret password, there's not some secret decoder ring. It just goes right through the door and into the cell, and it acts directly on the genetic material inside that cell in its nucleus. It acts directly on the DNA. And then sends instructions out of the nucleus into the cell so that you can create something new. Now that's awesome as far as our own steroid hormones, but you got to think about this. This is why steroids are so potent, especially in a medical setting, because if you start taking steroids for something, those steroids, the reason why we give steroids for medical purposes is because they are so direct. They will walk right through the cell membrane, enter into that cell, and do what they've got to do right there, right then, right now, as soon as they reach their target cell. This can be good, and then sometimes this can be bad. That's why you always want to know the side effects of having to take some type of steroid, because a steroid is not a living organism. It is just a chemical. It is a lipid-based chemical, and it's going to go through the cell membrane of any cell that it encounters, including any target cell. So we may give you a steroid so it can target specific types of target cells, but it may inadvertently go to some other target cells that we don't want it to talk to and talk to those cells and cause side effects that you cannot control because that steroid is already passing through that phospholipid bilayer of that cell membrane and going directly into the cell's nucleus and acting on its DNA and creating something that we didn't want. The second type of chemical messenger that falls in with the lipids are eicosanoids. And eicosanoids, they're just like hormones, except they act locally. That's why I have that taxi down at the bottom of the picture. You know, when you take a taxi, you take a taxi in your city. Usually when you go to take a taxi, well, you know, if you're out in like Mississippi, you can get a taxi to take you from one town to another town. Generally, taxis, you know, they'll drive you to some pretty far out places, but, you know, generally taxis only go but so far. A taxi's not going to take you from Georgia to Florida. That's not going to happen. He's not going to leave Atlanta, Georgia, and drive you all the way to Jacksonville, Florida, and turn back around. That's not what he's going to do. That taxi's going to stay his butt in Atlanta. All right. He's going to be local, and that's what an eicosanoid does. An eicosanoid, it's a hormone, but it acts locally. It acts in the local tissue. So it doesn't try to leave the actual tissue that it was released in. So if it was released in the liver, it's not leaving the liver. It wants to stay in the liver, and it wants to stay probably in that particular lobe of the liver and do what it has to do. What's funny is all four classes of these eicosanoids you'll learn about later. If you're taking anatomy and physiology as a two-parter, you won't probably talk much about these prostaglandins, prostacyclins, thromboxanes, and leukotrienes, trienes in your first portion. You'll probably take it in the second part of anatomy and physiology. These are very specific local acting chemical messengers. Prostaglandins are chemicals that I actually got a chance to do some research with, working with large animals, and how prostaglandins have a direct effect on reproductive cycles and whatnot. These thromboxanes, you talk about them when you're dealing with blood clots. It's great that they don't work anywhere else but that local area, because you want to make a blood clot in that spot to stop bleeding in that area. You don't want it to go any further. Some other lipids that you'll talk about throughout anatomy and physiology are things like glycolipids. Glycolipids are basically lipid molecules with some sugar molecule attached to it. Usually you talk about it when you're dealing with the plasma membrane or the cell membrane. And you'll see these again when you start talking about things that bind to receptor sites. You'll talk about that with glycolipids. And then, of course, your fat-soluble vitamins, vitamin A, E, and K. You recognize these like vitamin E. Vitamin E is easy to remember as a fat-soluble vitamin due to the fact that vitamin E is usually in a capsule, and when you poke a hole in it, it has that greasy consistency to it. And here's a picture of a steroid right here. See, there's those six carbon rings, and then there's that 5 carbon ring there. And that's what they mean by rings. They have these rings, and then they have the extra portion of the molecule sitting there as well. Okay, so that took us through lipids. That took us a minute to get through that. We've got carbohydrates. Carbohydrates are sugars. Carbohydrates usually throw people off for a loop because when they hear carbs, they think sugar. And when they think sugar, they think that bag of Dixie crystals they got in the cabinet in the kitchen. But they don't realize that carbohydrates have 3 different classifications. You have monosaccharides, disaccharides, and polysaccharides. When you're talking about a molecule, an individual unit of a carbohydrate, the functional unit of a carbohydrate is a monosaccharide. It's one unit of a sugar molecule. When you stick two of these units together, you get a disaccharide. And when you stick more than two together, you make a polysaccharide. When I'm talking about a monosaccharide, I'm talking about glucose. When I'm talking about a disaccharide, that's when you're talking about lactose. Um, when you're talking about a polysaccharide, that's when you're talking about a baked potato. That's when you're talking about starch. So you have different levels of these carbohydrates, and so you've got to know, you got to kind of keep that in mind. Okay, I'm going from small to really big. Now, when you eat sugar, when you're eating a carbohydrate, if you're eating a baked potato, it starts as a polysaccharide. That's how it begins. So you're looking at a polysaccharide, and now your body, your digestive system, its goal is to break that polysaccharide down to a monosaccharide so that your cells can ultimately use it. Your cells can't use a polysaccharide. They can't use that. That's too big. And even though they could use a disaccharide, they prefer not to, because in order to utilize that disaccharide, they got to break it down. What your cells really want is they want a monosaccharide. That's that random free battery just sitting on the table that they can use. So when you look at this image here, it shows glycogenesis and glycogenolysis. Or genolysis, or it depends on how you want to pronounce it, but we learned it as glycogenolysis. So, remember, lysis means to break down. So we take this glycogen molecule, which is stored sugar. This is how your liver and your muscles store sugar. They store it by taking an individual glucose molecule and attaching it end to other glucose molecules, and then they sit there, and then you break it down, and you snap off monosaccharide by monosaccharide, and you can use them, or you can put them together through glycogenesis and then store the thing all together. So some types of monosaccharide, you've got glucose, fructose. Generally, like sperm cells, they use fructose as energy. The average cell in your body uses glucose. Disaccharides is when you take two sugar molecules, two monosaccharides, and you stick them together. So sucrose, which is table sugar, that's your Dixie crystals. Lactose, which is milk sugar. Maltose, which is what we call bread sugar. Maltose is what you got in your beer or in your slice of bread. Lactose is in your milk. So when someone tells you they're lactose intolerant, what that means is that they lack the enzyme to split that disaccharide in half into two individual monosaccharides so that your cells can actually use them. Being lactose intolerant has very little to nothing to do with the fact that you don't like drinking milk, but you can eat an entire gallon of ice cream by yourself. That's not being lactose intolerant, folks. Lactose intolerant means that literally ice cream, milk, cheese, the whole gambit, all of your dairy products, your body lacks sufficient lactase enzymes to break down lactose so that you can maximize that sugar, take that disaccharide, split them into two monosaccharides. And then polysaccharides are when you got three or more sugars bonded together, and generally polysaccharides that we usually eat and find in the wild are very large. So, glycogen is what we find in animals because we have glycogen in our muscles, whereas starch and cellulose is a lot of what we eat. Starch is what we find in the baked potato. Cellulose is what you find in a piece of celery. You don't really break down cellulose well at all. That's why it's considered a non-digestible substance. We call it fiber or roughage. It's used to help you digest other food. You don't really digest cellulose itself. Uh, Nucleic acids. Nucleic acids are macromolecules that allow us to store and transfer genetic information like DNA and RNA. Now, DNA you can find it in two different places. You can find DNA in the nucleus of a cell. You can find DNA in the mitochondria of a cell. Using DNA from the mitochondria is a lot easier to manipulate than the DNA inside of a nucleus. It's just the way that the thing is packaged and it's wrapped. As a matter of fact, if we want to know, we found that maternal DNA, that DNA that's in the mitochondria, we found that that DNA in the mitochondria tells us a lot more about where we come from than the DNA in the nucleus. We can trace your mitochondrial DNA all the way back to figure out exactly where your ancestors exactly came from on this planet. Now, that doesn't make nucleic DNA obsolete. Perish the thought, because trust me, when you mutate nucleic DNA, that's when you can create cancer cells. So we don't want to do that. So both groups of DNA are very important. RNA is found in the nucleus and in the cytoplasm of the cell. And RNA, there's different types of RNA. Like messenger RNA is one of the ones that's most common. We'll actually talk about that later when we get to the cell chapter. Messenger RNA actually allows you to be able to take copies of the genetic material inside your nucleus and then bring it outside of the nucleus into the other area of your cell out in the cytoplasm and allow ribosomes to read it and then convert that genetic instruction into actual protein so you can build things. So RNA and DNA are actually pretty cool. And you can see the difference between RNA and DNA. You'll notice that DNA is on the right. It happens to be a double-stranded helix. It looks like a ladder that's twisted. And you'll notice how you've got the nucleotide here made up of a phosphate group. There's a deoxyribose sugar molecule. And then there's the nitrogenous base right here. And these guys go on to connect to a similar structure on the other side. And then they bond together in the middle with these hydrogen bonds. We can actually unzip this double helix. And when we unzip the double helix, that's when we're able to make copies of this genetic material to take into the cytoplasm of the cell and use that genetic material as instructions to do other things. The thing over there on the left, the RNA, this is a perfect example of some messenger RNA where we come over and we can connect that messenger RNA to a particular side or strand of the DNA, and we can allow it to make a copy of that material and take it outside of the nucleus. Another new type of nucleotide is ATP, which is energy. ATP is a magical molecule because it actually starts off as ADP, adenosine diphosphate, and then we attach a third phosphate group to it, which turns it into adenosine triphosphate. And that third P is what makes ATP important, because when we attach that third phosphate group, when we attach that third phosphate to the molecule, by attaching it, we trap energy within that bond. And so when you snap that third P off, the energy is released and the cell can actually use that energy to do different things. So what winds up happening is it makes ATP a very lucrative molecule that we can use anytime we want. All we have to do is continue to produce ATP, but in order to produce ATP, we actually need some of the other organic molecules to make that ATP so that there's enough ATP for the cell to run. Proteins are last but not least. Trust me, the reason why proteins are last on this list and the reason why there's very little about proteins in this chapter is because you could teach an entire semester on proteins and still not have enough time to talk all about proteins. I think there's like, I forgot how many different molecules we've we've learned about, different things that you can make proteins out of. I think there's like several thousand different things that your body makes out of protein. It's ridiculous. Think of proteins like Lego blocks. And so basically, you can connect these Lego blocks together and you can actually think of it this way. All right. Proteins are made out of these units called amino acids. And think of amino acids as Lego blocks. Because I can take just about any protein in your body and break it down to amino acids. And as long as I have my hands on those amino acids, I can take those amino acids and I can build an entirely new protein. You can have a protein that you use to carry things around in your bloodstream. I can take that same protein, break him down to his basic amino acids. He's just Lego blocks. If you've ever played any of the Lego video games like Lego Star Wars or anything, you already know that, there'll be things that are made out of Legos and then you destroy it and then you use whatever, if it's the Indiana Jones Legos or Star Wars Legos or whatever it is, you use your abilities in the game to turn those Lego pieces into an entirely different thing. It was, a spaceship. Now you took all the Legos and used it and turned it into a robot. Same Legos, same blocks, totally different structure. That's a protein. Protein is made out of the same amino acids that you used to build something totally different yesterday, but now you're using those same building blocks to build something entirely new. So it can work as an enzyme, and enzymes work as catalysts to help speed up metabolic reactions inside your body. It can act as defense because you use proteins to make antibodies to protect your body. It can aid in transport. We make these things called carrier proteins, which are proteins that float around in your blood to help you carry things like iron around in your blood. They contribute to structural support. You use proteins to help you build things like collagen fibers in your skin to help make your skin strong. They cause movement, which you use protein to make things like actin and myosin in your muscle cells. And when your muscle cells contract, it uses actin and myosin so that it can make you move. They perform regulation. You can use proteins to make special hormones. Yes, you do make hormones out of protein. And so you can use these special protein hormones to regulate any number of different functions in the body. And it provides storage. You can use these protein molecules, like, for example, hemoglobin is made out of protein. And you use hemoglobin to store oxygen in red blood cells. So this is a short list compared to everything that protein actually does in your body. It's amazing what protein does in your body. And so, when people say, oh, well, I don't believe in eating protein. Well, hold on a second. I'm not saying that you got to eat meat, but your body needs protein from somewhere. I know a couple of people, just a few, who are true to life vegetarians, some of them vegans. And they've been doing this vegetarian and vegan thing for a long time. I know a vegetarian he's been a vegetarian for 34 years, and he knows what he's doing. I've got a friend who she's a vegetarian. She's been a vegetarian for I'd say about 25 years. And I know a vegan who's been a vegan for probably about 20 years. All right. And they've talked to me countless times in the past about how they have to make sure that they get the right macromolecules in their system, and they're very healthy and they're doing very well and they look great. The thing about it is they make sure they get their protein in some shape, form, or fashion. You have to have protein, folks. Look at all the different things that you use protein for. So, just like lipids, the basic functional units of lipids were fatty acids for the most part. for like triglycerides. Well, for triglycerides, the basic functional unit was fatty acids. In carbohydrates, the basic functional unit is a monosaccharide. And for proteins, the basic functional unit, or the monomer, is an amino acids. And we have a total of 20 of these guys in living organisms. And they connect to one another through a bond called a peptide bond. So when you hear about a peptidase, that's an enzyme that breaks peptide bonds. And here's an image right here. You can see a pretty, here's a general amino acid here. It's not a specific type of amino acid because every amino acid has an amine group and a carboxylic acid group here. The thing that makes an amino acid unique is this R group. You know, all 20 of the amino acids have a different R group, some kind of weird thing that attaches right here. For some of them, it's like a carbon and 3 hydrogen that just attach here, just a, you know, this real basic group. And then others have this really long, extravagant ring-shaped structure that attaches here for its R group. If someone tells you that they're looking at an oligopeptide is just a strand of amino acids anywhere between 3 and 20 amino acids long. A polypeptide is pretty big. It's 21 amino acids to 199 amino acids. When you get up to 200 amino acids or more, you are no longer dealing with a peptide. You are dealing with a protein, full-fledged protein. Now, 205 long amino acid protein is a pretty small protein, but you can get to some really gargantuan proteins, and those guys, they get so big it's difficult for you to move them around within a cell. So what you wind up having to do is break that protein down before you can get it into a cell. And then one last thing before we move on to these last images is a glycoprotein, which you'll learn about those later on. Glycoproteins, we often refer to glycoproteins as markers on the surface of a cell. A glycoprotein is like a license plate. We refer to some of them, like the ones that is talking about here, this example, saying glycoproteins on erythrocytes or red blood cells. They determine your blood groups or your blood types. We refer to these glycoproteins as antigens. And so these antigens are on the cell surface, and we can see which ones these are, and that'll tell us what your blood type happens to be. These last images are just kind of talking about what we said earlier on. You can see here, this one is for the lipids. And you can see where there's this triglyceride, and the triglyceride was being stored in adipocytes in adipose tissue. And then you can see where if we take that triglyceride out and break off, if we separate the glycerol backbone from it, from the fatty acids can be released into the bloodstream, and they can float around in the bloodstream. Your cells will pick these fatty acids up and then use them as energy. If it's a phospholipid, remember we use the phospholipids in the cellular structure. Oftentimes we use it to create the cell membrane or the plasma membrane of the cell. Most of the time we use it as a double layer. We have that layer and that second layer, and it helps insulate and protect the cell from the outside environment. And then we have steroids, which easily pass across the cell membrane, and these steroids can actually be used as a hormone, the same as an eicosanoid, where these eicosanoids can then be released and utilized. And except the difference between an eicosanoid and a steroid is a steroid can be released into the bloodstream, and that sucker can travel from your toe all the way up to your brain. It can still function. An eicosanoid, if it's released in your toe, it's going to function in your toe. Probably the furthest that it would go is your foot, and then it'll deactivate. Carbohydrates, we can see this glucose molecule. There's numerous different things you can use with the glucose molecule. You could actually attach it to numerous other glucose molecules and store it as glycogen over here in the muscle tissue, or you can take that glucose molecule, drop it in the bloodstream, and the bloodstream will take that glucose molecule so that it can be used by a cell. that cell can use that glucose molecule and that fatty acid along with some oxygen in a mitochondrion to pump out some hard-earned energy. This molecule here is actually an ATP molecule, and so that energy is then transferred to that third bond of that third phosphate group over here so that we can use that ATP to use anywhere we choose. And then the last guy, second to last guy, excuse me, I'm sorry, is nucleic acid. Remember, nucleic acids and nucleotides, we're talking about DNA, RNA, and ATP, specifically DNA and RNA. Notice that the DNA is located either in the nucleus or it's located in the mitochondrion, while the RNA, this particular RNA, is located here in the nucleus, and it can actually exit the nucleus and take a copy of this genetic material from the DNA outside the nucleus into the cytoplasm so that you can read this information and create something. And then the last one is protein, and the protein, as I told you before, can be used for a number of different things. As you can see here in the image, this is figure 2-25D, and when you look at this picture, if you zoom in on it a bit, you can see what the protein is being used for. It's being used for numerous different things. Notice that it's the most diverse organic molecule because proteins can be broken down into amino acids, and amino acids can be used for so many different things at any given time. Well, my time is up, so I'm gonna have to end it right here. Hopefully we answer some of your questions. Hopefully that stuff will make sense. If you got any questions, you can leave them on the site, leave them on Google Plus, or you can leave them on the YouTube channel. We'd love to take your questions and do a couple of vids on that. We'll probably start getting some guest instructors to kind of come on and use their witty inventions and everything to kind of talk about this stuff a little bit more. So with all that being said, I'm out. Peace.